Thank you very much. It's an honor to be here, and I would like to thank Dr. Seçkin and Endo Foundation for inviting me to this wonderful conference, and also it's very meaningful that today is International Women's Day, and Happy Women's Day, everybody. I want to get started with, I know I'm going to speak about endometriosis, but we've done a pretty good job in our lab to define the functions of stem cells or progenitor cells in uterine fibroids. Basically, we demonstrated this concept that when you look at this entire tumor, what you see is like 95 to 99% of this bulk is made of these like terminally differentiated cells, and they die off. The real action is here in the stem cells. Although they make 1 to 5% of this entire bulk, they are the ones who proliferate and renew, but they lack progesterone receptors. So progesterone acts on these differentiated cells. They send signals like rankle and Wnt, and that makes these stem cells nurture. And as a result, the entire tumor grows in a symbiotic way. And we successfully targeted these signaling pathways, for example, to show that this could be exploited. I just want to put this picture out there. This is peritoneal endometriosis, rectovaginal nodule. As you can see, most of these lesions are made of primarily endometrial stromal cells. There's some epithelium in some of them. Here, there is no epithelium. It's mostly blood-containing macrophages and stromal cells, and tons of fibrosis. And we demonstrated that these stromal cells are the culprit of inflammation, the major pathology in endometriosis. And Tamer gave this picture to me, and I think he showed this picture again. Sort of, it's very compelling that retrograde menstruation. This picture was taken at the time of menstruation of a woman, and it's quite compelling that retrograde menstruation and the travel of these cells back here play a role. And in fact, until today, I was agnostic when I first started investigating endometriosis, but it turned out that all of our molecular findings show that retrograde menstruation probably accounts for like more than 95% cases of pelvic endometriosis. And then these three papers came one after the other. From this Chinese paper from Li, from Shi's and Tamer's paper in New England Journal of Medicine, and finally the Suda paper from a Japanese group. What these people showed is that both in utopic endometrium and in endometriotic lesions and in ovarian cancer, all of these epithelial cells contain the same mutations, KRAS, PI3 kinase CA, and ARID1A. It's very interesting. It's very compelling. And we kind of hypothesized that maybe the mutagenesis can happen because of inflammation and estradiol. And I also want to emphasize here that when you look at endometriosis, the mutations occur in the epithelial cells, right? And these groups also, they sequence the stromal cells. There are no mutations in stromal cells. So we've been working on stromal cells for 25 years. What happens in stromal cells is there are like tons of epigenetic abnormalities, problems with DNA programming. As a result, these abnormal transcription factors come up, and then these abnormal transcription factors give rise to prostaglandin formation that causes pain, tons of estrogen formation, And so it's important to recognize that, yeah, these cells from the uterus, epithelial and stromal cells, go back in almost all women. In 10% of them, they cause these symptoms. If they go to the ovary, now there is evidence that because they are the identical ovarian cancer driver mutations, they are involved in ovarian cancer. But although it was a negative finding, it was like extraordinarily important. Like she and Tamer's New England Journal of Medicine paper showed that if they end up in the rectovaginal nodule, they don't cause cancer. So there must be an environment conducive to cancer in the ovary. In fact, I wrote an article about this in Endocrinology recently, and one of the reviewers said, I want you to speculate. You know, they sent a reviewer back, they said, this is too lame, I want you to speculate, what do you really think? And I said, fine. So we think that estradiol is the culprit because there's like tons of estradiol made in the ovary, maybe measured in 10,000 picograms per ml. And estradiol could be converted to catechol estrogens or quinones that go directly to DNA and depurinate DNA and cause accumulation of more mutations. So usually one mutation doesn't cause cancer. You really have to accumulate a lot of mutations. This is pure speculation, by the way. I don't think anybody showed this. There are a lot of studies in breast cancer, but not in ovarian cancer, right? And I look for partners from this group. Maybe we can get together and study some of this stuff. Hugh has done a lot of valuable work in endometrial stem cells. He's going to talk about cells that come from the bone marrow. A lot of like progenitor local stem cell work has been done arguably by Carolyn Gargett from Australia. And Carolyn thinks that these stromal cells, mesenchymal cells that accumulate around the blood vessels may serve as like these progenitor cells that make the next endometrium in the next cycle. And these cells exist, again, both in the functionalis and basalis. And probably there are more of these in the basalis. I mean, sometimes the thing speaks for itself. I mean, there's nothing left other than basalis and maybe some islands of functionalis, as Larry suggested. But these cells these tissues must have something to do with rebuilding the endometrium, even if they incorporate some other bone marrow cells. And I think this was a Leyendecker hypothesis, and he never wrote it somewhere, but you know, it makes sense. If the separation occurs at a more superficial level, less number of maybe stem-like cells would be carried to peritoneal cavity. On the other hand, if it occurs in a deeper level, it may. I mean, this shows that a lot of cellular defects may cause the same phenotype. You can argue that maybe vascular cells could be the culprit. So epigenetics is everything about DNA programming is about the stem cells, and one of epigenetic regulation is how these CPG islands on DNA is methylated or unmethylated. And we demonstrated that this is like totally abnormal or different in endometriotic stromal cells compared with endometrial stromal cells, in that the distribution of methylated or unmethylated CPG islands in endometriosis, if they are functional, reside primarily in open seas. That means in the intergenic regions, uncharted territories between the genes. Still, this is not well understood. We published this some time ago. And we demonstrated that two genes that are alternatively methylated came up two transcription factors, GATA2 and GATA6. To simplify things, GATA6 is the good transcription factor that helps normal endometrium to decidualize properly. GATA6 is the bad one that causes a lot of inflammation. And we were interested more about this. And what we did was we went ahead and charted the entire genome. So in the genome, like, there are 3 billion base pairs, right? And 25,000 some genes in the human genome. So these charts represents how GATA2 binds to the entire genome around these like 25,000 genes. And it looks like the green normal endometrium GATA2 binding is much stronger, whereas it's much weaker in endometriosis. On the other hand, we see I want to show one more thing for this retrograde menstruation hypothesis. I think this kind of supports it. This yellow stuff is utopic endometrium from patients with endometriosis. As you can see, sort of like, this is like genome-wide, not a single gene. It kind of falls in between. So it kind of shows that there's something wrong with the utopic endometrium of patients with endometriosis. And this is GATA6. And again, it's like really prominent in the genes upregulated in endometriosis. It binds more avidly, and less avidly in both in normal endometrium and utopic endometrium from patients with endometriosis. So that relationship is not so clear here. This is like how, if we put it all together, it sounds like there is another level of complexity. These are the histones. So there is DNA methylation, right? Then there are like the histones around which the DNA is like organized. And there are like certain residues on these histones that are methylated or acetylated. And so it looks like in normal endometrium, there is a GATA2 binding dominance. And for the genes upregulated in normal endometrium, also the enhancer type of histone modifications are promoted. In endometriosis, exactly the opposite takes place. GATA6 is more prominent, and endometrial type genes are suppressed, whereas GATA6 causes the activation of more pro-inflammatory genes. And again, we are going to come back to this concept just a little bit with, when we talk about stem cells, just a little bit. When we overexpress GATA6 in normal endometrium, when we ectopically express it, it shows that it changes the phenotype of normal endometrium to a more inflammatory type by overexpressing these like 30 genes from the immunome, which overrepresents the upregulated genes. I think Tamer invited me to talk a little bit about this subject mostly. Human iPS cells, induced pluripotent stem cells. Like in 2006, or a little bit earlier, Yamanaka, who's a Nobel laureate now, who's an orthopedic surgeon, showed that if you add like four simple genes, overexpress them in any cell, like you can make a skin biopsy, or you can get a bone marrow biopsy, and simply add 4 genes, 4 transcription factors, this erases the entire DNA programming, that epigenetic programming, and make them pluripotent stem cells, which are called iPS cells. And a lot of investigators, they were very successful to differentiate them into cardiac cells or lung cells or liver cells, but they could never be differentiated into the endometrial type cells. So that was our contribution. So the story is like back to your embryology classes in medical school. iPS cells are kind of like embryonic stem cells. They could be differentiated into primitive streak and intermediate mesoderm earlier, and then they were differentiated into kidney cells, but people never were able to follow the other line to differentiate them into endometrium through coelomic epithelium to Mullerian duct to endometrium. So what we did was we applied this Wnt beta-catenin enhancer together with some of the other factors, first to take intermediate mesodermal cells that originated from iPS cells and differentiated them to coelomic epithelium, apparent by these key markers like PDGF receptor and TCF21 and some of the others, as you can see both at the mRNA and protein level. And the next step was to turn coelomic epithelium to Mullerian duct, which was a big deal from day 6 to day 8. And again, we demonstrated this by showing these markers. But the punchline was, I think, we all wanted to see HOXA10 and HOXA11, right here, of course, and the progesterone receptor. So that, I think that was the key. I don't think people were able to, differentiate these cells ever to a cell type that express a steroid receptor. And we managed to do that. So there was like a prominent, respectable expression of progesterone receptor. Once we use this, now we added a AZA treatment, which is a DNMT inhibitor, together with E2. And this KIR compound is the key. So it inhibits GSK3 beta, which activates beta-catenin. And we all know that Wnt system is very important for uterine development. And as you can see, very respectively, we demonstrated both PRB and PRA, both by Westerns. And also, we demonstrated HOXA11 protein and PR protein. And these are all nuclear, by the way. And finally, I think this is the last data slide that I'm going to show, is we decidualize these cells. So we use the time-honored decidualization hormonal cocktail made of estrogen, medroxyprogesterone acetate, and the cyclic KMP analog. So now we are at day 14. We treated them for 8 days. And in each one, we use like 2 clones, like of human cells. And these are like XX, 46XX iPS cells. And as you can see, like at least in this clone, IGF-BP1, which is the ultimate marker for decidualization, went up by 1,000-fold. In this one, it was almost like 100-fold. So there was some difference between the clones. And FOXO1, prolactin receptor, these all were, and we demonstrated IGF-BP1 by an ELISA. And so, again, I mean, we are talking about probably technology that will be developed over the next 20, 30 years, because iPS cells were intended to use to cure human disease. And the earlier trials were started in Japan, I guess in honor of Dr. Yamanaka in macular degeneration. And they made retinal cells from a different patient and put it in the patient, a different subject and put it in the patient suffering from macular degeneration. And it was complicated by a change of law in Japan, and also I think right now they start to see some problems. So by any means, this is not perfect yet, but we can use our imaginations just a little bit, and we need that for endometriosis. And so defective endometrial stromal cells contribute to uterine factor infertility, endometriosis, and endometrial cancer. And again, for example, grade 1 endometrial cancer has a lot of stromal cells, right? If you treat these patients with progestins, as gynecologists, we all know that, they respond. Once you get to grade 3 endometrial cancer, they are like sheets of epithelial cells. They don't respond to progesterone. Why? Because there are no stromal cells that contain progesterone receptors. So you can use this technology for that purpose too. Replacement of abnormal stromal cells with normal ones could be a potential and novel therapeutic approach. Of course, we have to use our imaginations how we are going to do this. And again, we can reprogram them by these factors, induce pluripotency, use differentiation factors to appropriately make them progesterone responsive and put them back. And thank you so much for your attention.